Good afternoon and welcome everybody. Welcome to our final webinar in the UCCE and the UCIPM Biologics Educational Webinar Series. This series has been organized by Dr. Zhang Wang and Rachel Long. My name is Cheryl Reynolds and I'm with the UC statewide IPM program. Peter Casina is also here with us to run our poll questions and troubleshoot any technical problems that might come up. And so now I'd like to introduce our speaker today. Dr. Surendra Dara is an entomology and biologicals farm advisor with UCCE, San Luis Obispo, and Ventura counties. And today he's speaking on the topic of synergistic or antagonism, integrating crop biologics into IPM. So now I'm going to pass this over and you can go ahead and share your slides.
Good afternoon, everybody. Hope you can see my slides. Thank you, Cheryl, for the introduction, and thank you, Peter, for all the logistic support. And thanks to Zheng and Rachel for inviting me. The topic has two parts here. One is there any synergism among these biologicals with other management practices, synergism or antagonism, and how do we actually integrate biologicals into IPM? First, I want to compare the growth and health of humans with plants to give you a general idea of what is important for us to be healthy, and the same way what is important for the plants to be healthy. You know, the genetics play a critical role. In humans, we cannot choose certain traits like we do in plants. Although, you know, when we select our companions, we might be using some of those, like a phenotypic, like the looks and intelligence, some of those things. But intelligence comes after, right? Like mostly it is looks to get us into trouble. And then when it comes to plants, you can look for high yielding and disease or pest resistant traits and so on. So that is the start for either for plants or humans. Then the environment that we grow, like the socioeconomic conditions, family setup, and those play a role. Whereas more or less it is the same, the environment, the soil, and other environmental conditions, and the kind of resources that the farmers have, and what kind of knowledge and financial resources they can use to do certain things, all influence the crop growth. Then we have the nutrition, which is similar in both humans and plants. You know, you have major nutrients and then supplemental nutrients that come from different sources, and then overall health. You know, exercising, eating healthy, and taking care of our regular health checkups, and having these probiotics, like which are beneficial microbes that promote our health, because we have trillions of microbes in and on our body. So we are actually not a single organism. We are we are a team. And then we have other things too to maintain our health. The same way the plants also have these biostimulants, and health checkups can be compared to regular monitoring and using these biologicals that I will discuss, and all those things. So if you look at human health, you just don't depend on one thing. There are several factors that influence our growth and development and health and how we maintain it. The same way plants also use all these cultural practices, the genetic makeup, variety selection, and proper nutrition, irrigation, and then regular crop protection or other health management. So this is what IPM is. And then what are biologicals in agriculture? So this is the definition I have, or my definition, that biologicals can be any biotic, which have living organisms, or abiotic, or maybe there could be dead or live organisms, or some non-living or dead materials. These could be like chemicals or some toxins extracted from plants or other organisms, but they have to be of this biological origin, that are used either for crop production purposes or protection purposes. And then here we have these different categories: biological control agents, biopesticides, biostimulants, soil amendments, And then we have these non-pesticidal beneficial microbes and biochemicals, which are pheromones, and then mineral-based materials. And these are some, a brief overview about the mode of action of these different categories. Biocontrol agents, mainly either parasitization or predation. Then biopesticides, depending on the kind or depending on the active ingredient, it could be multiple. It can be like a contact poison, or it has to get into the target site and then cause infection. Then we have biostimulants, mainly to induce systemic resistance in the plant, that is, build the plant immune system and other beneficial natural processes. Then soil amendments. These are added to improve the soil structure and biological activity, like microbial activity. And as a result of that, they promote plant growth and health. Then we have these non-pesticidal ones like mycorrhizae and other ones that improve nutrient and water absorption. Then these pheromones used for mating disruption, very successful in some cropping systems. And then mineral-based ones, depending on the active ingredient, it could be a pesticide or it could be a biostimulant. So we have these are general mode of actions for different groups of biologicals. And then I will go over each one again. So parasitoids are basically insects that parasitize other insects. So it is not like parasite, it is a specific term used here in entomological terms. So they are parasitoids. We know several examples of those wasps. And then again, we have several species of predatory insects and predatory mites and other arthropods. So there are different approaches here. Either you conserve what is there in the field, you don't have to release or do anything. Whatever is there, you want to protect it by choosing certain sprays that don't cause harm to them, or providing additional food sources, a refuge, and so on. Then you have this classical biological control approach, which is primarily used for invasive pests. So you bring natural enemies from the native area of an invasive pest and release in its new home. Then we have augmentative biocontrol, where we periodically release these natural enemies. And we have several examples of them in greenhouse production or open field production, like strawberries or nut crops. And these are used as a part of IPM program by regularly releasing them based on the target insect or target pest. So this is how biocontrol is used: conservation, classical biological control, or augmentative biocontrol. Then we have different categories of biopesticides, either based on microorganisms. They have live microbes, or they have metabolites from these microbes, or they could be botanical extracts or other plant-based materials, or they can be these peptides and various organic molecules, polysaccharides and so on, or they could be mineral-based. Now we will look at them one by one. So if you look at microbial, insecticides, and acaricides, there are these different categories. These are called entomopathogens. We have bacteria, fungi, microsporidia, nematodes, and viruses. So bacterium, what you are seeing here is, this is the Bacillus thuringiensis, a very popular bacterium used as a biopesticide. And this is the toxic, the protein that produces this crystalline toxic protein is actually inserted into corn, soybean, cotton, and other crops. The gene responsible for that is inserted into these crops, so that when insects feed on these crops, the toxin works, and rather than the infection, it directly kills the insect. So how do they actually infect or attack insects? So when we look at bacteria, they have to be ingested, and this crystalline protein is activated under high pH conditions of the insect gut, and they make holes in the intestine and then eventually kill the host. Then fungi, when they come in contact with the insect or mite host or arthropod host, They produce these germ tube. They also release enzymes. They get inside the insect body, multiply, kill the insect, and then emerge, like they produce more spores, and then they spread the infection there. Then microsporia are related to fungi. They have to be ingested, and they cause infection and kill, and then again they're released into the environment. Nematodes are microscopic worms. They don't actually kill the insects, but it is the symbiotic bacteria they carry. Like each species of these are two different popular genera, Steinernema and Heterorhabditis. Each of them has a particular genus of bacterium that they host, and that when these infective juveniles of these nematodes go out, look for the insect host, they enter through natural openings. Then they release these bacteria, or the bacterial activity actually kills the insect. Nematodes multiply inside the insect body, and then when it dies, and then when they complete 1 to 3 generations, they come out and search for new hosts. Then there are two kinds of these viruses. One is a nucleopolyhedrovirus, and then a granulovirus. They need to be ingested by the insect as well, just like bacteria or microsporia. They need to be ingested. And nowadays, everybody is familiar with what virus does. Virus basically requires that genetic material from the host. So they get that material from the host to multiply, and basically they liquefy the host dead insect, and then again that cycle continues. As you can see, each one has a specific mode of action, and the way they enter or cause infection either through multiplication or by toxin, right? So this information is important for us to know to choose the right organism or right product for our particular need. Then microbial fungicides. There are bacteria. Many of you are probably familiar with these Bacillus amyloliquefaciens and other species, Cetylis and Streptomyces and so on. And then we also have Coniotherium militans, this fungus. This is and Trichoderma. They are they're very effective against some plant pathogens. They can either antagonize because of those microbial metabolites, or they parasitize, or basically they colonize that area, so there is no room for these pathogens to infect the host. So that's how they work. Then microbial nematicides. We don't have a lot of species here, but some Bacillus species and Burkholderia, Sepacean, and then fungi, especially this fungus, Purpureocilium like lalacinum. This is commercially available. The other ones are also available, not everywhere, but we have this in the US. This is available. And these are, again, either through antagonism, like directly microbial metabolites kill the nematodes, or they parasitize. Some of them, some fungi actually form the rings and they constrict, they strangle these nematodes. So that's how these microbes attack other ones. Then there is another mode of action, is ingesting the spores of this fungi, and then they cause infection there. So this is how bionematicides or microbial nematicides work. And then we have molluscicides. These are mainly for snails and slugs. And there is one nematode, Phasmarhabditis hermaphrodita, and then spinosad from this bacterium. This is Entrust or Radiant, and some of those commercial products that have this active ingredient. And then there is also research that shows that the extract from tea oil, camellia, that this is actually saponins. They are also, they have molluscicidal activity. Then microbe-based materials. They don't have the live organisms, but they are derived from beneficial microbes. Then we have these two examples, ivermectins, you are familiar with abamectin, which is kind of derived from ivermectins. So this comes from this bacterium, Streptomyces avermetilis. Then spinosad, as I mentioned, it is also from another bacterium. These are produced through fermentation, and then they have this nematicidal or insecticidal properties. Then we also have these two products based on Burkholderia rhinogenesis and Chromobacterium substitugate. These are also fermentation products, but at the end of the fermentation, they're killed. These live bacteria are killed by heat, and this product contains various metabolites and basically these dead bacteria. So it is the metabolites that have this insecticidal property. Then we have a huge range or a wide variety of botanicals, anywhere from azadirachtin that comes from neem tree to essential oils, and then if you go down the list, you have several are available in the US or California are commonly used. And if you look at this one, chrysanthemum, like pyrethrin from chrysanthemum is very popular. And then, then terpenoid compounds and other ones are also very popular in various production systems. So we have several botanicals because, plants and every organism has some chemicals or some proteins or carbohydrates or other materials that have various properties. And these are examples of those alkaloids, flavonoids, and terpenoid materials with insecticidal or biostimulant properties. Then we have these organic compounds. So blad is a polypeptide that comes from the germinating sweet lupin seeds. Then this spider venom peptide, you're familiar with that one too. It is a new class of mode of action, recently introduced, recently added active ingredient. These two are peptides that have, this is a fungicide, this is an insecticide, then this is a polysaccharide. Chitosan is a polysaccharide from the shells of the shellfish, exoskeleton of the shellfish, and it is a fungicide. And it also has biostimulant effects, properties. Then mineral-based, diatomaceous earth. It is basically fossilized, powder of fossilized diatoms. These are microorganisms. What they have is silicate. Then we have sulfur and other minerals, either used as fungicides or acaricides, or as a soil amendment, for soil amendment, or as a for other purposes. Then we have mineral oil, and this is also an insecticide or fungicide. So these are all different categories of biologicals that we can use either for plant growth or for plant protection. Then I just want to bring your attention to this comparison, like how these abamectin, pyrethroids, neonicotinoids, and spinoterem are produced, or how they are developed. When you have these natural molecules that have fungicidal or insecticidal or acaricidal properties, you look at them, and then you have to increase the scale of production to a commercial level. And when you can synthesize, it increases the, sometimes you improve the stability of the product in the environment. And also it is easy to produce on a commercial scale. So that is how these synthetic analogs are developed, but they act either exactly the same or more or less similar in their activity. And then it is important for us when we are trying to use biologicals. As you can see, there are a lot of categories here. And then some are microbes, some are metabolites, some are, like they seem to be just carbohydrates or protein materials, but they have a complex interaction with everything in the environment. The environment in the sense that like the temperature, humidity, and all those things, or pH of the soil or water used could influence, or how they are attached to plants, and how they interact with plants or other microorganisms or pests or diseases and everything. So they have very complex system, the way they interact with everything in the crop canopy. So it is important for us to understand the mode of action and how they work and what works best under different conditions. Then it also helps us to know that some of those can be biostimulants or fungicides or insecticides. So it helps us to understand these properties so that we can take advantage of them and also avoid applying certain pesticides or other materials. If this particular one does 2 or 3 things, then we don't have to apply the other ones and reduce the cost. So now I want to mention about induced resistance in plants. That is how the immune system is basically triggered in plants when they are exposed to various stress factors. So the first one is when plants come across these either herbivores, insects, or mites, or pathogens, or some chemicals. And sometimes it could be a virulent, which means like some pathogen that can actually cause the disease, and sometimes it is avirulent. It is related to a pathogen species, but it is not, or maybe a subspecies or a variety or a race that does not actually cause infection. Okay, and then whenever they come across these stressors, biotic or abiotic, there is a particular pathway, a particular mechanism that is triggered. Genes related to the production of salicylic acid and certain proteins related to this infection are activated. As a result of that, they produce this acid and these proteins. And this kind of defense mechanism is called systemic acquired resistance, SAR. So this is when they have pathogenic pests or pathogens and chemicals and so on. Okay, so this is mostly like harmful ones. But when plants come across these beneficial microbes, these are like, the mycorrhizae or other microbes that are in the environment, they don't cause any harm, then another kind of pathway is triggered, because plant sees that there is something on me or something is interacting, and probably it is taking some nutrients from me, but not causing any harm other than that I provide nutrition, it provides protection or something like that. So then there is another kind of mechanism that is triggered, producing this jasmonic acid and ethylene, and also pathogenesis related proteins. And this one is called induced systemic resistance. So we have one systemic acquired resistance when their plant is exposed to stress, real stress factors. Then the other one is when plant is exposed to the beneficial ones. So this is like taking medicines or something like that, no, it is like antibodies being produced in our body when we have cold or when we have a health issue. And this one is like taking those probiotics, yogurt, or there are other products that supply these beneficial microbes. And so this is the difference here. And we can use this, however, this particularly this beneficial microbes and other ones, we can use it to improve the plant health. So here are some examples. We have a variety of microbiologicals that are used for pest control, disease control, and soil health and biostimulation. So here we have these bacteria, fungi, nematodes, and viruses. Then here we have these fungi, different species, and then these genera of bacteria that are used for disease control. Then, this something like this Bacillus, Pseudomonas, you keep seeing it because there are multiple species and different strains have different kinds of roles. Some could be just biostimulants, or some could be antagonizing another pathogen, a pathogen, and that rate you apply also makes a difference. At a very low rate, something could be just a biostimulant. At a higher rate and frequency, it could be a biofungicide. So it is important to understand, and that is why also these have different labels as a biostimulant or a biofungicide and so on. And as you can see, trichoderma, these are fungi. And then for biostimulation, you have all these Bacillus, Pseudomonas, and then Beauveria bassiana, which is an entomopathogenic fungus. This actually can be used as a biostimulant or as a disease antagonizer. And then this one is yeast that we have here. So these are just the active ingredients based on microbiologicals. And the next slide is all kinds of biopesticides. So this is actually, I was working for the past few days to put this together. Looking at a lot of literature and the information I have and so on. So what you see here is all these arthropod pests, and then you have various kinds of diseases, then nematodes, snails and slugs, and then different kinds of weeds. And these are bacteria, fungi, nematodes, viruses, botanical materials, or toxins, or metabolites, and others. So you can see different categories, and there is a very big list depending on the target pest, and depending on the source. We have a lot of them, and several, except for just a few, most of them are available in the United States for weed control or controlling nematodes, snails, or slugs, or pests, or diseases. So compatibility. Okay, this is an important part that we need to focus on, because we know that these various groups or categories of biologicals are available, but we want to know how they are compatible. So before that, I want to just show some details here. No interaction. Okay, you can add them. You can be adding a biostimulant, or maybe use a biofungicide or bioinsecticide, but they have no interaction with other inputs, so chemicals or other materials, or even other biologicals. So product A and product 1 have no interaction. So 1 and A are entirely different. So it is not an issue there. Then that means they are compatible there. Then there could be additive effect, which means when you add one and another one, you end up seeing the total of those two. Like, for example, you used a biofungicide, which provides 30% control, and then you used a chemical fungicide, which provides again another 30% control. Then you have 60%. That is how it works. Then there is a synergism. Here, this 30% and 30% does not result. Individually, they provide 30 and 30, or 40 or 40. But when you add them, there is a synergism that increases their efficacy of the combined materials. So it could be more than 2. It is when 2 or 3 or 4. So that is a synergism. Then antagonism. This is incompatible. When you add 1 plus 1, then the result is less than 2, or it could be 0. Some combinations could be very, very bad. It makes the other one inactive or ineffective. So this is very important to understand, and the manufacturer's guidelines or labels are critical to, very important to go over those details. And each application situation might be different. So sometimes if the information is not available, you may have to contact the manufacturer and obtain that information. These details were not previously very widely available, but several biological producers are now making it available with more research and more information, and the quality has improved, and all these data and other details are also available now. Then we want to understand the interaction of these biologicals and what are the influencing factors. So we have these 4 main categories here: biocontrol agents, biopesticides, biostimulants, and other agronomic inputs. I just put them all together. And then we have these non-pesticidal biochemicals, mainly pheromones and so on. So looking at this, when you are using biocontrol agents, it is important to see how they interact with pesticides, or when you're applying pesticides, we need to make sure that they are compatible. There is no harm. This can be biological pesticides or chemical synthetic materials. And it is also important to know how when you have a fertilizer spray or some other materials you are putting in, and is it going to impact the nematodes that are entomopathogenic nematodes in the soil, or anything that you have applied as a foliar spray. So it is important to understand those combinations. And then mechanical tools. If you are using a vacuum and if you have natural enemies, the mechanical tool which is the vacuum is going to remove those natural enemies. But you have to weigh the benefits and then those disadvantages, which is important for the situation and so on. And the factors that influence are the source and choice, right? You need to get your biocontrol agents from a good source, and also the choice means using the right natural enemies for the right target pest, and then handling them. Sometimes you have to store them overnight, or the way they are transported and the way they are applied, that is also very important. You don't want to expose them to harsh conditions. And sometimes you may be able to provide some additional food sources, unless it is a specialist natural enemy feeds only on the pest. Some of them feed on other pests or pollen or other nutrient sources. You might be able to provide them so that you can sustain their populations. Then the environment, when you are releasing, or is there any environmental impact, a sudden increase in temperatures or something like freezing temperatures that would affect. So these conditions are very important for the success of these materials. So we need to monitor them. And then again, the time of release. When are we releasing? Is it too early when there is nothing, or too late? Or how are we releasing in patches or spot treatment, or all over? And then rate of release too. Like, are we releasing 100,000 or 30,000 or 10,000? These all influence its success. Then biopesticides. Then they could have interaction with other ones. And some biopesticide combinations have synergistic impact, and sometimes these biopesticides could have negative impact too. So we need to understand. And then other ag inputs, just like the previous category. Then the water quality. Some have probably a neutral pH, or some might work well under acidic pH or alkaline pH. So we need to adjust the water quality as needed. And then more or less the same kind of the choice of the biopesticide, whether it is a botanical or microbial, metabolite based or microbial one. Then what is the mode of action, and am I choosing the right one? And then am I using proper storage and handling protocols to keep them alive until we apply? And again, what kind of environmental impact it is going to have? Did I apply when it is too hot, or is it like too late or too early? The same thing, because some materials work based on contact, so you want to apply them at the time when the insect or the target is there. Sometimes some actually, like entomopathogenic nematodes, they can go around, look for their host. So all these things play an important role there. Then biostimulants. Biostimulants are generally compatible with the other agronomic inputs, but if you again have a biological material, like if it has live microorganisms, then it is important to understand how they interact with other agricultural inputs, and what is the impact of water quality, and again soil conditions too. That also is important. The pH and other conditions, like the sandy or clay structure, and other factors of the soil influence that. Then the purpose and choice. What purpose are we using this for? Is it to increase the nutrient absorption or water absorption, or is it for protecting from high salt conditions or drought conditions? So you have different biostimulants are these beneficial ones based on the purpose. So we have to choose the right one. Then same as the other ones, storage and handling, and then the impact of the environment. Those that are in the soil may not be influenced as much by the temperature and humidity, but I mean by temperature, but the humidity, like the water conditions and soil moisture and other things, could be influencing the success. Again, time of application. So this is also important, and then rate of application. A lot of times you have to realize that how much are we applying, and what is the inoculum level of pathogens in the soil. Like if you have some pathogens that are using a biofungicide to apply to the soil, you have to realize there could be millions of the pathogens, whereas you're applying just thousands of these beneficials. So the rate of application, distribution, and how it is spreading, and what kind of requirements it has to multiply in the soil and spread, and so on, all those factor influence. Then biochemicals, again, these are mainly pheromones I'm referring to, and they are compatible with other control options, because they are mating disruption. They're used for mating disruption, and sometimes they are used as attractants too. Not just mating disruption, they are attractants. You can use them with baits, or just if it is a mating disruption, you just use with pesticide application or other control methods. Then here too, timing and the rate that you either release or apply, or the number of units you put in a unit area, all these are important for the success of them. So here what we see is the first 3 points, or the first few points, are the things we have to watch for, what we are mixing, or what the kind of interaction they can have. And the other ones are also our decision, how we use to increase or improve their success. Okay, so here basically you need to have good knowledge, and it also, the success also depends on your budget, and what kind of logistics issues you have when you can apply, and is it too late or not, and so on, and then how well you can implement these. Okay, and then what are different use strategies? You can use as a seed inoculation, apply to the soil, or treat the transplants, apply to the soil, or use as a foliar spray throughout the crop production, depending on the material we are using and the purpose it is used for. And then another one is Beevectoring, which is using these pollinators to deliver microbial pesticides, or maybe we end up using them to deliver other pesticides too, I don't know. But these trichoderma and some other these microbial fungicides, and even Beauveria bassiana kind of insecticides can be used. There are some containers or devices attached to these hives. So when these pollinators exit these hives, they pick up this material, and when they visit the flowers, or basically the crop, they will be distributing the fungal inoculum or whatever they're carrying all over the target areas. So this is very effectively used. And the question you may have is like, wouldn't they impact the health of these bees? Usually they distribute all that material during their foraging. So when they go back into the hives, they go through a different route. So they don't go through and take that pesticide inside. And then hive temperatures are usually detrimental to these microbes. So they are generally not affected. And this technique is very effectively used in many areas. And we also have some commercial devices available, these hives and other ones available for Beevectoring in the US. And then a few examples of compatibility of biologicals. There is so much research about these, so we won't be able to go over all those details, but I just want to give a general idea. Like, you can use multiple entomopathogens. That means you can use fungi with bacteria or fungi with nematodes, And there has been a lot of research done, like combining different entomopathogens. You can do the same thing with the biofungicide materials too, bacteria, and actually you do have some products, not as biofungicides, but as biostimulants that have multiple species of bacteria and fungi. So, and again, entomopathogens with natural enemies. You might think these would harm these natural enemies, but they have used these fungi and predatory mites and fungi and lady beetles and other natural enemies successfully. There will be interactions though. That is why it is important to understand. Some could impact these natural enemies and some have no impact. Actually, there was this additive effect for pest control. Then using entomopathogens with pheromones or attractants or netting, even spraying entomopathogenic fungi on the nettings, and and then that can provide control. So it is like a physical control along with microbial control. Then using pheromones with pesticides, and that is very common in many systems. And then biopesticides with the chemical or botanical biopesticides, in the sense, microbial ones with the chemical or botanical pesticides, then biopesticides using pollinators to deliver the biopesticides, and biostimulants with the synthetic fertilizers. This is an important area. When you add these biostimulants, you can reduce the amount of these synthetic fertilizers, because these biostimulants improve their absorption or bioavailability. So there are various ways of using them, and these can be like, again, biostimulants with pesticides or other materials, combinations or rotations, and various ways at different stages of crop production we can use them. And then we are towards the end of this presentation. And what is the scope of biologicals in IPM? If you think biologicals, where do actually biologicals fit? We have all these host plant resistance, cultural biocontrol, behavioral, and all of them. I want to show that the transgenic crops are not exactly like a biocontrol tool, but I want to use that as an example because we are using this microbial protein, toxic protein, and put it in the plant, so it is related to it, okay? That is, but then if you look at everything else, we have biologicals in every category of IPM, right? If you look at cultural control, all these biostimulants and all the materials you add for agronomic purposes there, then biocontrol agents, obviously, then behavioral control using these pheromones, or using these biopesticides with the attract and kill strategy, and then nets treated with biopesticides, then microbial control, different kinds of fungicides and insecticides we looked at. And then chemical control, these are like chemical molecules obtained from plants or microorganisms. So we see biologicals everywhere. We do use them, whether we realize it or not. We do use them everywhere, and they can be used everywhere too. And then what biologicals do? Summarizing, like improve the plant vigor, and they impart resistance, or sometimes they compensate for the pest damage by increasing the vigor or production of this improved nutrient absorption or production of foliage and so on. Then they reduce the crop loss as a result, or if it is a biopesticide, by killing the insects, or biofungicide reducing the disease, they reduce the crop loss by controlling them. And then they also are very important for resistance management. That is an important part of IPM, because repeatedly using any tool, whether it is biological or synthetic material, sometimes or a lot of times they can lead to resistance problems. So we want to use all these and reduce the risk of resistance. Then mechanisms of insecticide resistance and for biologicals. So this is I'm not going into the details, but I quickly want to bring your attention to these various mechanisms, how insects or arthropods develop resistance, either by altering their behavior, reducing the penetration of the target, or the pesticide, or changing the target site, or getting rid of it, increase the sequestration within the body, or some metabolic changes that make the toxin or pesticide less harmful. And sometimes having these symbiotic microbes in their gut or in that, in that system, that also imparts resistance. And this is the case with both the synthetic pesticides and some biopesticides. So it is important to remember not to overuse anything. Then finally, some strategies. How do we use biologicals? As you could see, there is a lot of information, a lot of categories. It is important to understand what it is and choose the right ones for the right purpose, and especially of good quality. And then again, we have to develop the strategies now that we know all these materials are available. We can pick and choose what fits my budget, what fits the crop situation or the target pest or disease, or if it is for a biostimulant for economic purpose, we need to look at all those and use which one would be more successful. And again, storing and handling, and following all those guidelines to make them, to keep them viable and maintain their efficacy. And then, it is also important to combine and rotate, not just to these biologicals. Any tactic, IPM has several control options. Again, starting from host plant selection to various cultural practices, even before we have to spray or do something about pests. Changing agronomic practices can play a critical role in pest suppression. So we can use these biologicals or synthetic materials along with those tactics. At the same time, we need to monitor the system, not just for the pests, but what is happening when I am doing this. It could be a positive impact when I'm continuously applying a particular one. It could be a positive thing, or it could be causing other unknown or unexpected harms. Like with there are a lot of resistance problems with the spinosad and pyrethrins. The same way that those interactions, maybe it is controlling the pest, but it is also doing a lot of harm to natural enemies. So we need to look at those. And, again, that resistance development from biologicals is also an important thing to monitor. And then cost is usually, or for several biologicals, cost is a concern. Like efficacy has improved significantly over the years, quality and efficacy, and now we have all this information about how to use. But at the end of the day, some biologicals could be expensive compared to synthetic materials, but we want to protect the available options. We want to make sure that the available effective options last longer. So which means we want to prevent overuse, and sometimes adding these biologicals into our crop production can have a longer impact. Not just a short term, it might be expensive, but in the long run, it evens out, and then the positive benefits could be, or the benefits could be much more than the initial investment. So these are various strategies. So the conclusion is biologicals are critical tools for sustainable agriculture, and they are an important or integral part of both IPM and insecticide resistance management. And it is very important to have a good knowledge of these biologicals and various strategies, their mode of action, and use them effectively for their success. I have this earlier version of this this presentation and other presentations on my YouTube channel about the IPM and other things, and this is the particular one. Biologicals in IPM is there if you want to see, but I don't have all the information I presented here in that one. And then this is about insecticide resistance management. The image I showed you, there is a lot more detail there. So you might want to subscribe or watch my videos there. Well, that is the end of my talk, and let us see what questions you may have.
Okay, and so right now we just have one question. So if anybody does have questions, please enter them in the Q&A, and we'll go ahead and ask those. So this first one, and I guess it's the only one, it's kind of a longer one. So biologicals, particularly biopesticides, usually are developed and commercialized by big companies targeting high-profile crops, meaning mainstream crops which mobilize high levels of economic resources, either by price or scale. Many minor crops lie outside industry interest and are not registered because the investment is not paid off by expected revenues. So the question is, do you think that this niche could be taken by small farmers, cooperatives, and stimulated or fostered by government?
Okay, this is an excellent question, but there are a couple of issues here. As far as I know, a lot of these biologicals are not produced by big companies. It is only now the major companies are getting into expanding their portfolio to include biologicals. Until recently, it is all smaller companies. Then the crops, I am not quite sure what you mean by major or minor. If you look at these corn, soybean, cotton, these are like a huge crops, but biologicals are not used as much because there is no, it costs more there. Then if we are talking about minor crops, which are specialty crops, that is actually where biologicals are effectively used. So I want to clarify those two things. Then your question of the last point is, if there are these farmers cooperatives to have their own. I know this happens in other countries, Mexico and some other countries, they do have it, which is good as long as things go well. But what if there is an error? Someone accidentally, there is a contamination with a pathogen or something else, and which, the QC is not there, like a big registered company would do, that is the risk. We might be spreading a pathogen there. But if there is a system to ensure that it is done correctly, maybe several years ago I was also thinking of along those lines, something for the farmers to produce and use. But afterwards I realized that there could be there are potentially several risks also.
Okay, so that is actually the last one we had in the Q&A, and I don't see any in the chat. Surendra, if you're still there, there is another question if you have.
Yeah, I'm just looking at it.
Okay.
So how can we tell if the biologicals we purchase receive are still viable before applying? That's a good question. And, it is usually, it is the company's responsibility to ensure, and if it requires refrigeration or special transportation conditions, they do take care of that. And but if something happens in between, that is probably related to the logistics issues. But they do assure that the quality and the number of units or number of propagules that are present in a milliliter or 1 gram of that material, or how many live ones there should be. It is all according to the guidelines and requirements. So it should be done. If everything is handled correctly, it should be viable.
Okay, I think we have covered everything. So I think we're going to go ahead and close out. So thank you everybody for participating in this last webinar. And thank you again, Surendra, for presenting.
Thank you. My pleasure. Bye, everybody.
Thank you. Bye.